After watching this video, you should be able to use compound microscope. Follow my instructions, rewind if you have to, and you will be ready to go on your own. There is a strict rules how to transport microscope. You have to grab it with your right hand by the arm of the microscope. The left hand has to be at the base of the microscope. This is the only correct way to transport it from one place to another. Never grab microscope like this. This is very wrong. So right hand at the arm of the microscope and left at the base of the microscope, and you're ready to go and bring it where you have to. Working with the microscope, you must be always seated. Your eyes should be just over the ocular lenses. Never observe the image standing up. Unwind the electric cord of your microscope and plug it into the electric socket. Make sure that the cord does not hang from your station. Having the electric cord hanging from the workstation is not safe and considered to be violation of safety in the laboratory. Before you start your work, make sure that the microscope stage is lowered all the way down. We move microscope stage up and down using the coarse adjustment knob. Here you see fine adjustment knob, but we do not want to move it yet. Fine adjustment knob we use only when view specimen under high magnification, not low magnification objectives. When you move this knob, microscope stage move very little. In fact, so little that you can barely see it moving. By using these knobs, we control distance between the observing specimen and objective lens. Now let's make some observation, answer some questions, and make notes. When you turn the coarse adjustment knob towards yourself, does the stage moves up or down? Look. Note that each ocular lens of the eyepiece has a magnification power of 10x, meaning each ocular lens magnifies image 10 times. How many eyepieces do you see on this microscope? Besides ocular lenses, microscope has objective lenses with various magnification power. The lowest power of objective is the smallest objective with a red stripe around it. It gives a magnification of 4x. Together with an ocular lens or lenses, the total magnification power is 40x. 4 multiplied by 10, 10 is magnification of ocular lenses, equals 40x. So the total magnification of microscope when you use 4x lens is 40x. Now you know how to find magnification of microscope. The total power of magnification of the final image formed by combined lenses is a product of the separate powers of the two lenses. This is important to keep in mind, since every time you would draw a diagram of a specimen, you have to indicate total magnification either under or above of that diagram of yours. Most often we start microscopy with this objective. This objective has the lowest magnification power, and therefore it allows you to find image that you are looking for more quickly. It is called scanning objective lens. Please remember this. Do not forget, this lens has the lowest magnification power of your microscope. Every time you finish working with the microscope and prepare it for the storage, you have to make sure you turn the scanning objective lens to working position. Now tell me, what is the magnification power of the objective with the yellow stripe around it? It is 10x. Now, by analogy with previous calculation, calculate the total magnification power of the microscope when you use objective with yellow stripe around it. Pause the video if you have to. The correct answer is 100x. Like scanning lens, 10x objective lens is considered to be low magnification objective lens. Now tell me, what is the magnification power of the objective with the blue stripe around it? You can see it. It is written on the objective. 40x. And what will the total magnification of the microscope be when you use this lens to observe a specimen sample? 400x. You take power of objective lens, multiply it by power of ocular lens, and you get 400x. So you just learned that the magnifying powers of objective alone ranges from 4x to 40x. In a separate video, I will show you how to use oil immersion objective with magnification power of 100x. We use this lens in microbiology courses most of the time. An international color code has been devised to make it easy to identify the magnification of an objective lens. A colored stripe that corresponds to this code encircles modern objective lenses. The code is black, corresponds to 1x. Brown 2x, red 4x, yellow 10x, green 20x, light blue 40x, dark blue 63x, and white corresponds to 100x. It is on an oil immersion objective. Now let's see what will happen if we will put letter E under the microscope. In this video, I will not cover how I will make slide with letter E. Usually students are given already prepared slides for them. Note how to hold slide without leaving a fingerprints on it. Our fingerprints are oily and we do not want to make our slides dirty. The slide goes on the stage of the microscope and it is secured in place by mechanical stage that has a clip. This is a really cool part of the microscope. It can move your slides to the right, to the left, towards yourself, away from yourself. All of this you can do by turning two knobs labeled here as a mechanical stage control. You can turn them in any directions you want to.
Here is a mechanical stage control that has two knobs. If I move the upper knob, the stage moves. It moves towards you. Or you can move, if you move this knob away from you, it's gonna move away from you. So towards and away. If I take the lower knob of the mechanical stage control, this one, the stage moves left or it moves right. Really cool device.
But before you put your slide on the stage and secure it in mechanical stage, first you have to make sure that microscope stage is all the way down. Remember, we are not looking through the ocular lenses yet. When we secure a slide in the mechanical stage, we have to make sure that the scanning lens in the working position, meaning perpendicular to the stage of the microscope. You should hear a click when the lens is in its proper place. Now you have to bring microscope stage all the way up. You are not to look through the ocular lenses when you do it. You are to look from the side when you move the microscope stage all the way up. You have to see when to stop so you don't scratch the expensive lenses with your slide containing the specimen. You also might break the slide if you do not see objective pushing against it. You know how to bring microscope stage up. You learn it on the beginning of this video. You turn coarse adjustment knob away from yourself. And when the stage of the microscope is all the way up with the letter just under the scanning objective lens, you may look now through the ocular lenses. In this course, you will always have to start with the scanning lens first and later move on with the other objectives. If you had difficulties to follow me so far, here is the realistic video how to position slide on the microscope stage.
Here's how you put your slide into the mechanical stage control. There is a clip here, as you could see. So you move the clip, you put the slide all the way there, you let the clip go, and here it is. Now your slide is in the position. Note, we're still not looking in the ocular lenses. We're looking from the side. What we want now is to bring microscope stage up. And looking from the side, we want to make sure that roughly the image we want to look at is under the scanning lens, not out somewhere on the left or right. Quite often students move it to the right and say, I don't see anything, right? You have to make sure that it is right under the scanning lens. Then you are ready to look through the ocular lenses at your image. Now you move slowly, course adjustment knob towards yourself. When you do this, the microscope stage moves down. Another hand could be on mechanical stage control, so you can center your image. It is very important that image is centered in the field of the microscope, in the field view of the microscope.
Why it is important to center the image you are interested in, I will tell you in a bit, but now let's see what we have. Are you excited to see the letter under the microscope lenses? Here we look. And we see nothing. What happened? Why is it so dark? Oh, we plugged the microscope to the electric socket, but we forgot to turn the power switch on. So here it is. Now it is on. You can see the light coming through the light source. What is that? We see very small dark area. This area is supposedly called field of view. Usually it is about of this size, so you can see large area of the specimen, in our case a letter E. But do you? What do you think happened? Why the field view is so small? You see, under the microscope stage there is a light source. The size of illuminated area is controlled by the field diaphragm. This diaphragm could be opened more by field stop diaphragm ring. Let's turn this ring to open up the field view. Wow, that's much better now. We see more. Compare what we had at the beginning and what we have now. Now we can see much larger area than at the first time. Apparently one of the students before you was moving field stop diaphragm ring and left it partially closed. But we opened it up. Good. We will never use this part of the microscope during the course, so please always leave this open, so students in other class after you will not get all confused. Okay, let's move on. I'm still not happy with the final image though. Don't you think it is too dark? I certainly think so. The reason it is so dark, it is because the student before you left another diaphragm closed. This diaphragm is just under the microscope stage. On the microscope stage, there is a substage condenser. The substage condenser gathers light from the microscope light source and concentrates it into the cone of the light that illuminates the specimens with uniform intensity over the entire field view. It is critical that the condenser light cone be properly adjusted to optimize the intensity and angle of the light entering the objective front lens. When we work with a living specimen, a microscopic organism or living cells, we want the substage condenser to be closed with the iris diaphragm lever. Do you see this small knob just under the microscope stage? Why do you think we want substage condenser to be closed? Because the living organisms or living cells are transparent, and if there will be too much light going through them, you will not be able to see them. Another reason is you perhaps do not want to kill them, because light is heat. Tiny life organisms or living cells will be like on a frying pan. You might not feel it, but they certainly will suffer, be damaged, and die. But when you work with a non-living specimen, you want this light, because the slides you will work with are fixed and stained tissue or fixed killed microorganisms. They are not that transparent. They are stained. And this is the reason why you want your substage condenser to be open. So here is our image with the closed substage condenser. Here is our image with partially closed substage condenser. And here is what we want, open substage condenser. We open it with the small knob called Aries diaphragm lever. But I'm still not happy with the quality of my image. It is blurry. Do you see how blurry it is? To fix this problem, you have to move stage a bit down while looking through ocular lenses. To move stage down while looking through ocular lenses is normal. This is how we find the right focus. While observing the image, move the coarse adjustment knob very slowly. Here it is. Letter is now in focus. This is how we want it to be, nice and sharp. Now it's time to diagram what you see. Do not forget to indicate the total magnification of the image. I hope you remember how to do this. Do you remember the lens we have been using? It was the scanning lens, the lens with the red stripe around it, correct? The magnification of the scanning lens is 4x, but you do not want to enter this number. You want to write the total magnification, that is 40x. As you remember, we put letter E on the stage of the microscope as we read it, correct? So what happened? Why the letter now looks upside down and backwards? The letter appears upside down and backward because of the lens system. This means that the slide must be moved in the opposite directions that you want the image to move. If you want to move it to the right, you move the mechanical stage to the left. If you want image to the left, you move the mechanical stage to the right. Let's see this in action. Now we will change magnification from 4x to 10x, which is higher. Still, we consider this magnification as low magnification. And this is how the letter looks now. You can see and diagram more details such as paper fiber and the rough edges of the letter. You still can see the letter, but it occupies the most of the field now. Do not forget to record the magnification. By the way, what is it? The objective lens is 10x. Therefore, the total magnification is 100x. So the image is magnified 100 times.
And now we're going to change lens from 10x to 40x. Total magnification of the microscope is going to be, look what happened. You see? The objective lens is hitting the slide. We don't want this to happen. So we have to lower the stage. That's why it's important to look from the side when you change objective lenses. Now we can move to 40x lens safely. And of course, center our image. Before we look at ocular lenses, make sure the image is under the objective lens.
And here is what we see. We do not see the whole letter anymore. With increase of magnification, there was decrease of the field of view. But look at the fiber of the paper the letter is printed on. The fiber is a cellulose fiber. Diagram all the details of this view. And do not forget to record the magnification. 40 multiplied by 10 will be 400x. The letter magnified by 400 times. And you know now the reason why we do not see the whole letter anymore. With increase of magnification, field decreases. See here all we observe. With increase of magnification, the field decreases, isn't it? Why do I repeat myself like a broken record? Knowing this has a practical value, because this means every time we move from low magnification to higher magnification, we have to center the image or we will lose it. It is not going to appear in the field of view. Here we have the image that is not centered. Look what will happen when we go from 4x lens to 10x lens. The letter E is left out of the field. This is why it is important to center the image before moving to a higher magnification. Here are 3 field views of 3 human tissue samples. Choose one to diagram. It is very important to draw in biology classes. Remember, you do not have to be an artist to do so, because biological drawings are not about artistic expressions. They are about observations. To draw is to observe. After you finish your work, we turn the rotating nose piece on the objective lens that has a red stripe around it. We call it scanning lens. Microscope always have to be stored with a scanning lens on, working position. Now, then we load the stage. I am lowering the stage, as you could see, and we remove the slide. You turn off the microscope and you take the electric cord and wrap it around the microscope. There is a strict rules how to transport microscope. You have to grab it with your right hand by the arm of the microscope. The left hand has to be at the base of the microscope. This is the only correct way to transport it from one place to another. Never grab microscope like this. This is very wrong. So right hand at the arm of the microscope and left at the base of the microscope, and you're ready to go and bring it where you have to.